Super Lab. In this Super Lab, we're going to investigate how to determine the experimental conditions that would produce specific results. Before we get started on the experiment itself, let's go through some background information, which will outline for us a description of the skill in a little bit more detail.
Most SIN skills ask you to think about how the methods used in experiments impact the outcome of the experiment. SIN 504 requires you to pick out which methods will give you a specific result in an experiment. These questions often ask you, how could you get, and then ask you how you could change original experiments to get a specific result. Before you can figure out how to change an experiment, you need to be able to figure out what the original methods used in the experiments are.
Exactly. All right. So you can expect to see a lot of phrases like, how could you get blah, blah, blah? Or, how would you change such and such experiment to get blah, blah, blah? All right. That's the format of a lot of these style questions. All right. Let's take a look at a passage that is going to highlight these questions for us. The first paragraph of this, you'll notice, is a little bit of background information about the experiments that are going to be run. The second paragraph outlines the first experiment that was run. The third paragraph outlines the second experiment, and the last paragraph outlines the third experiment. So let's get started by reading the first experiment. As we read through the first experiment, there's going to be a lot of terms that are going to come up right away that are going to be really tricky. In fact, you might even have trouble pronouncing some of them, especially because some of these words are Latin names of organisms. So just try to stick with the vocabulary as we go along. One tactic that we always use in these is to circle, highlight, or underline any words that you are confused about as we go along. So we can go back and find them later so that you are not confused about the vocabulary as we do this paper. All right, so with a pen in your hand, armed with the ability to highlight, circle, or underline any words that you hear that are confusing, we will have the background information about the passage.
Enzymes catalyze, speed up the rate of biological reaction without being consumed in the reaction. Pyrocosis, spiriosis, an organism that lives in the ocean thermal vents where the temperature remains near 100 Celsius, produces the enzyme protease. Protease catalyzes the breakdown of proteins to release the amino acid choline. An enzyme's activity is measured of its effectiveness for a given reaction, expressed as a percent of its maximum measured effectiveness.
All right. That was a mouthful. There was a lot here. So let's first go through and circle any words that are tricky to us right away. Does any word jump out at anybody that they circled that we wanted to find?
Pyrocosis.
Pyrocosis, spiriosis. Nice. Any other words that jump out that we need to make sure that we clarify? Prolidase. Excellent. Any other words that jump out that we should go through? All right, one I just want to add in here is catalyze. Probably a review word based on your knowledge of chemistry from last year, but we should go over it just to make sure we know it. Any other words that jump out? All right, so as we start to take a look at these three different vocabulary words, remember that there are three major ways that you can learn new words. They are either defined for you in the passage, that's one way. The second way is either to use context clues or background knowledge to try to figure out what the meaning of the word is. And the third way is to look it up. In ACT, we can't look it up, so we're going to try to avoid that way as much as possible. Let's get started with our three words. The first word is catalyze. Is it defined in the passage? Do we have to use context clues, or should we look it up later based on this word?
In the passage.
Excellent. Where do you see it in the passage?
Right next to it. Speed up.
Perfect. In parentheses, you'll notice that this is the definition for the word catalyze. All right, so when an enzyme catalyzes a reaction, it just speeds up that reaction, makes the reaction happen faster. All right, in this case, we're speeding up a biological reaction. All right, the next one, pyrocosis spiriosis. All right, that is a very funny sounding name. All right, all this is a Latin name of an organism. All right, Latin names are something that we're going to cover in a few months in class, but essentially what all it is the specific scientific name for an organism. For example, we could also read it a word Homo sapien. All right, that is our species Latin name. All right, the short version of that would be human. All right, if you see it in italics, and actually this is written slightly incorrectly because this S should be lowercase, you'll always see the first word capitalized and the second word lowercase, and they're both italicized. That's why you'll just know it's a scientific name of an organism. There's no real definition here. It's a species of bacteria. All right, just that's what that funny word is. Last but not least, prolidase. Prolidase defined for us anywhere? No, we don't see it anywhere. Can we use context clues or background knowledge to figure out what it is? No, probably not. As we start to then examine it, prolidase is an enzyme. All right, the way that you know if it's an enzyme is because it's a word that ends in A-S-E. Any word that you see in science that ends in A-S-E is an enzyme. All right, that means that it's doing something. All right, especially ones that end in A-S-E. You can then try to figure out what the start of it is in order to figure out what that enzyme does. For example, this one is prolidase. All right, and the prolid part refers to proline. So it's an enzyme because it ends in ase, and it makes proline because that's what it says on the front. So it's an enzyme that breaks down proline. I'm sorry, that breaks down proteins to make proline. And you can see that right here. Prolidase catalyzes the breakdown of proteins to release the amino acid proline. All right. Any questions about any of these vocabularies? All right, let's move on to the second paragraph, which is about experiment 1. We have a volunteer to read experiment 1 for us.
Many enzymes function only in the presence of a metal ion called the cofactor. 1 ml of a standard solution containing prolidase and 0.4 millimolar a unit of concentration for 1 ion will mix with 1 ml of a solution of protein X, of which proline is a component, at a pH of 7.0 and 100 degrees Celsius. After 10 minutes, a substance that reacts with free proline to produce a bright red color was added to the solution. The solution was diluted to 100 ml, and the intensity of the red color was measured and used to calculate the activity. The procedure was repeated for different concentrations of CO2 and then for the ion and zinc ions at all of the concentrations.
That was also a mouthful. All right, this is a long, long description of a fairly complicated experiment. All right, let's go through and make sure we have all the vocab down before we move on. All right, did anybody circle, highlight, or underline any words that we should clarify before we move on? Say one more time. Diluted, which is right here. Nice. Other words that we should identify? Any word that you think could jump out to be tricky later on. Manganese ion. Good. Any other words that we should discuss? All right, I'm going to underline cofactor. That's going to be pretty important later. Okay, any other one? I'm also just going to underline intensity. All right, let's take a look at these words. First, let's get started off with cofactor. All right, find in the text, context clues, or look it up. In this case.
Context clues.
Context clues? And so what do you think it means based on the context clues?
It's probably metal ion.
Excellent. In this case, it's the metal ion. And what does the metal ion do that actually makes it a cofactor? We take a look at that whole first sentence. Many enzymes function only in the presence of a metal ion called a cofactor. So what does the cofactor allow to function? Right, it's the enzyme. All right, the enzyme functions only in the presence of a metal ion. All right, think of it like a lock and a key. So if we have a cofactor, if we have an enzyme that looks like this, essentially we're looking for a cofactor, which is a little metal ion that will kind of finish the key for us. cofactors. And then what happens is the two pieces physically fuse together, which allow it to function. All right. So without a cofactor, the enzyme won't actually work. All right. More and more cofactor, more and more activity. Let's take a look at diluted. Can anybody give us the term diluted? We're going to have to use background knowledge for this. What does it mean to dilute something? Like if your Kool-Aid is really diluted, or your lemonade is really diluted, or your I think it'll be diluted. What does that mean? All right, so what do we mean by the term diluted?
It's when there's too much water or too much of something in something else.
Excellent. Yeah. So like your lemonade, for example, if it's too diluted, it's really, really watery. So there's either too much water or not enough lemon. Either way, you get diluted lemonade. It's real, like, not very flavorful, not very strong. All right. And then last but not least, this word intensity. What do we mean by the term intensity, especially when referring to the intensity of the red color? Yeah. So bright, like a real intense red. So it's red, or it's just, like red. That would be not very intense. But like red would be very, very intense. Right? Okay. Last but not least, manganese ion. This is just, manganese is an element. All right, it's one of the elements in the periodic table. All right, an ion is just different charged version of the element. All right, we have 2 more paragraphs left. Let's take a look at experiment 2. Who would like to read experiment 2 for us? Fantastic.
Experiment 1 was repeated, but CO2 plus held constant at 1.2 millimolar was a cofactor in all trials, and the temperature was varied from trial to trial.
Excellent. Does anyone have any words here that jump out at them that we should define before we move on? I want to just highlight 2. All right, what do we mean by the word constant? Kept the same. Fantastic. What do we mean by the word varied? Changed. All right, so in this case, CO2 was held constant this time, and the temperature was there. Right, the temperature changed. Last but not least, let's get through the last paragraph.
Experiment 2 was repeated, but from trial to trial, the temperature was held constant at 100 Celsius and pH was varied 2 to 3.
Excellent. All right, in this case, what do we keep the same? Temperature. What do we change? What was varied in this experiment? pH level. Great. All right, now with this exact idea, it's going to get us through the next section. When you have experiments like this, you should be able to fill out a chart that looks roughly like this. We have 3 different experiments in this case: experiment 1, experiment 2, and experiment 3. Now in each experiment, we control some things, we change some things, and we measure some things. This is a preview of a process called Pac-Man. That's a lot like Hulk that we're actually going to cover in a couple weeks in class. All right, so when we say control an experiment, what are we talking about with those things? Yeah, those are things that are held constant throughout the whole experiment. Nice. What about if we're changing something? That means we're changing the amount of it. And then measure just means this is what we're measuring in the experiment. So what I'd like to do is just fill out this chart quickly for each of these columns for every experiment. All right, so let's get started with experiment 1. In experiment 1, what are we keeping the same throughout the entire experiment based on our information here? So before we can really go through and maybe measure the control and measure the change in this experiment, we should talk about how this experiment actually works. All right, so let's pretend that we're in a lab. Here's a lab coat, and you're going to do the steps that are listed here. All right. In the end, in this experiment, what are we trying to measure about the enzyme? All right, what are we trying to measure about the enzyme? Yeah, the activity. And remember that we learned that the purpose of the enzyme is to break down proteins to make proline. All right, so we're trying to measure the activity of the enzyme. Now, how are we going to, and we can see that in this statement right here. It was used to calculate the activity. Now, how are we calculating the activity? Are we like counting the number of individual fluidases that shoot out somewhere with a little counter? How are we actually measuring how active this enzyme is? What are we looking at?
The intensity of the color.
Right. So in this case, the intensity of the red color is going to measure the activity. All right. Now, how is the red color being made? Where is the red color coming from? It comes from this sentence right here. The red color comes from a substance that does what?
Reacts with proline.
And it turns what color?
Bright red.
Bright red. So we're adding this substance that reacts with proline to make a bright red color. All right. So in the end, what was the purpose of the enzyme again? What did the enzyme make? Prolidase, the enzyme, makes what? There's a lot of keywords in here that start to get a little bit tricky. All right, so prolidase up here, it catalyzes the breakdown of proteins to release what?
Amino acid.
Good, the amino acid proline. So in this case, if the enzyme is working a lot, will it make a lot of proline or a little proline? Lots, because the more the enzyme works, the more proline is produced. If there's a lot of proline, and we add a chemical that reacts with proline to turn red, will it be really bright or really dim?
Right?
Really bright. Exactly. So the brighter the color, the more proline. The more proline, the harder the enzyme works. The harder the enzyme works, that's the activity level. All right, so if we look at this experiment, we're going to write a little flow chart. It's like we have the enzyme, which is releasing proline. The proline is getting added to a substance that makes it turn red. And so more red is more protein. More proline, which is more activity. Okay, so let's go back again. In this first experiment then, what are we measuring in experiment 1? What's being measured here? Good, the activity of the enzyme. So we can call it the enzyme activity. Now let's start to take a look then, what was changed in this experiment?
Can it be the different concentrations of fuel?
Good, and what do we call those metal ions? Those are referred to as what, that we looked at before, one of our vocab words. CO2 plus, Mn2 plus, and Zn2 plus were all different what?
Cofactors?
Yeah, these are all the cofactors. All right, so in this case, we're trying it at different cofactors. All right, and now we're keeping a whole bunch of stuff the same. What are we keeping the same in this experiment all the time? Right, that we're using 1 milliliter of a solution all the time. All right, 1 milliliter of a solution of prolidase. All right, what else are we keeping the same all the time?
Oh, 0.4 millimolar.
So now careful, this is a millimolar of cobalt ion. So this is one of the cofactors. So this is something we're actually going to end up changing. What else is kept the same? Good, the diluted solution. All right, with proline. So it's always the same amount of proline, solution X. And now what else are we going to end up probably keeping the same? If you were in a lab running this experiment, we want to keep what else the same?
100 millimeters of diluted.
Yeah, you want to keep this diluted solution at 100 milliliters. And especially then maybe even looking forward at some of the other experiments, what are some of the other experiments changed that this one is keeping the same?
Temperature.
the temperature. All right, so in this case, the temperature is staying constant. We have the 100 mils of the diluted solution. We have the 1 mil of prolidase, we said. All right, and there's probably one other thing that's important to keep in mind here compared to the other experiments that they're changing. All right, that this one is staying the same. Good, the amount of time we're looking at, that's going to be really important. I didn't think of that one. And then there's one other thing too. The amount, oh, that's CO2 plus. Yep, so some of them are going to use CO2 plus. What else is staying the same, especially looking at experiment 3? Like, what does experiment 3 change that this one doesn't? Yeah, the pH. In this case, the pH will stay the same. All right, let's jump on to experiment 2 then so we can get through the rest of these questions. In experiment 2, what are we measuring? Let's see. If we're repeating experiment 1, what are we measuring? Yeah, we're still measuring the enzyme activity. All right, but what are we changing this time? Yeah, and this time we're changing the temperature, but what are we keeping the same?
The CO2 plus.
Excellent. So we're keeping the cofactor the same, the CO2 plus. What else are we keeping the same?
Okay.
Good. We're keeping the pH the same. And what else are we keeping the same? Anything else? We're still going to repeat everything else from here. So all this other stuff that we're keeping the same here is going to be the same right here. So we're still using the 1 milliliter prolidase solution. We're still using 100 milliliters of the diluted solution. Time should be the same. All that should be the same. All right, let's just jump on finally to the last one then, experiment 3. If we read experiment 3 says experiment 2 was repeated. So what are we measuring in experiment 3? The activity level again. All right, but what are we changing this time?
The pH.
This time we're changing the pH and keeping what the same? Yeah, the cofactor is still going to be the same. The temperature is going to be the same.
The 1 milliliter of prolidase.
Yeah, the 1 mil of prolidase is going to be the same. Excellent. Another one? We're also going to use hopefully the same enzyme. We have prolidase that's in there, the same amount of protein, which is 100 milliliters diluted solution. All right, time hopefully is going to be the same as well. Okay, all right. At this point, what you should do is take a look at these tables, and it should start to make more sense now, what they're showing, because we know what's changing and we know what's staying the same in each of these. All right, table 1, what were we changing in table 1? All right, table 1 is experiment 1 in this case, and what were we changing? The cofactor. So what we see is the activity percentages for different cofactors. All right, how active was it? All right, what about table 2? What were we changing in that case? The temperature. So you can see the activity level for different temperatures. Last but not least, experiment 3, what were we changing? pH. So you can see the activity level for different pHs. Okay. Now, if you feel like you still want to work through some more of those questions, you can. And you can go back over them and re-listen to that section. We're going to move on. If you feel like you were lost at all, you should press pause now and rewind to listen to any of that. We're going to take a look at the example question. According to experiment 2, what temperature would you use to reach an activity percentage of 6%? All right, in this case, we're looking at experiment 2, which means we need table 2, because it tells us in experiment 2, which is right here, that we need to look at table 2. So we look in table 2, and we're looking for an activity percentage of 6%. Where would we find an activity percentage of 6%?
Between 40 and 50.
Excellent. In between 40 and 50 degrees Celsius, because you see 6%, here's our percent units, that would fall somewhere between 0 and 8 would be 6, which means if it's between here, it's also got to be between these two. So it'd be somewhere between 40 and 50, probably closer to 50 than to 40. So that's why the estimation there is about 47 degrees Celsius. Okay. At this point, you should pause and make sure that you can go through all of the guided analysis, fill in the blanks. What this section is doing is essentially giving you one quick review about what happens in every section of the reading. If you can fill in these blanks, you really have a good understanding of what happened in the experiment. If you can't fill in the blanks, that's a good sign that you should pause here again and go back and re-listen to the explanation for what actually happened in the experiment. So you should pause right now and work through guided practice questions 1, 2, 3, and 4 to make sure that you understand all three of those experiments, I should say. So press pause now, and when you're ready, press play to go through the answer. All right, so we're going to get started then. If you press play at this point, you have all of the blanks already filled in. All right, you should not press play until you have the blanks filled in, and we're going to go through the answers together now. All right, guided practice question 1 says fill in the blanks about the basics of the lab. That's coming from this right here, the first paragraph that you read in the passage itself. All right, it says the organism blank blank. All right, what we're looking for here is the name of the organism. What's the name of the organism that we're using in this case?
Pyrocosis furiosus.
Fantastic. All right, Pyrocosis furiosus should go right here. Pyrocosis furiosus. Excellent. Which lives in the ocean at temperatures around
100 degrees Celsius.
100 degrees Celsius. Right here. All right. Makes the what enzyme? What enzyme are we looking at?
Makes the prolidase.
Prolidase enzyme. Excellent. All right. You're going to notice that there's a lot of keywords in this. All right. Prolidase, protein, proline. It's important to make sure that you keep it straight. All right. Now the enzyme. What enzyme are we referring to again? Prolidase again. Breaks down what? Proteins, and releases the amino acid. Proline. You can actually pronounce it either way. All right. So right here it says prolidase the enzyme catalyzes the breakdown of proteins to release the amino acid proline. Perfect. This is the basics that you need to know about the experiment. Let's jump into experiment 1. Some enzymes only work when they're exposed to a substance called a what? Cofactor. All right, we got that right here. Many enzymes function only in the presence of a metal ion called a cofactor. All right, now this is the details about the experiment. Blank, 0.4 milliliters of blank, and 1 milliliter of blank were all combined at a pH of blank and a temperature of blank. Right. There's a lot to fill in here about this experiment. All right, let's start with any of the blanks that we can find. All right, the most obvious ones are probably temperature and pH. All right, so all combined at a pH of what? What pH are we looking at? 7, so a pH of 7. What was the temperature that we're using here? 100 degrees, oh, here it is, 100 degrees Celsius. Okay. What did we use 1 milliliter of? So that's this part right here. 1 milliliter of blank in this experiment. All right, 1 milliliter of a solution of protein X. So 1 milliliter of protein X. Nice. What did we use 0.4 millimolars of? Yep, 0.4 milliliters of cobalt ion, which is CO2+. And what are we missing from this whole thing?
1 milliliter
Of that contains what?
proline.
Prolidase. Good. So we need prolidase, the enzyme. Combine it together with the cofactor to make it work. Combine it with the protein it's going to break down at a pH of 7 and a temperature of 100 degrees. In this solution, you would expect the what enzyme? What enzyme are we working with again? You'd expect the prolidase enzyme, yep, to break down the what? Prolidase breaks down proteins, good, and releases the amino acid proline, or proline, either way. A substance was added that turns the color blank when it reacts with blank.
Red.
Turns red. Right. Let's see. It's right here. A substance that reacts with free proline produces a bright red color. So it turns color red when it reacts with what? Proline. Right. The brighter the blank color, the more blank released, indicating a higher blank level. Red color, the more what? Proline, right? Because that's what it's reacting with. So the more red, the more proline, the higher the enzyme's what? What are we measuring about the enzyme?
Activity.
Yeah, the activity level. Because that's how fast it broke down that protein into proline. All right, last but not least, this experiment was repeated using various amounts of blank, blank, and blank. Nice. Cobalt ion, Cl2+. What other ions?
Is it uphill with the
Yep.
14X?
Okay, so CO2+, Mn2+, Zn2+. These three substances acted as the, what did we say they're actually? Those metal ions are called what? Back at the beginning, we looked at it in A. The cofactors, nice. For the which enzyme? The one enzyme we've been working with all the time? Yeah, prolidase, good. Whew. All right. What about experiment 2? Experiment 2 is exactly like experiment 1, except blank at a concentration of blank was the only cofactor used. So what cofactor was used? CO2+. At a concentration of what?
1.2 millimolar.
1.2 millimolar was used, or was the only cofactor, and the what was varied in this case. Temperature was varied, right? Okay. And then experiment 3. Experiment 3 was exactly like experiment 2, except blank was changed and blank was held constant. What was changed in experiment 3? pH. And what was held constant? All right. At this point, you could reasonably go through and fill in all of those blanks. You know exactly what's going on in the experiment. All right. And that is going to make the rest of this so much easier as you start to go through these questions. If you couldn't do that, it's a pretty good indicator that you should go back and re-listen to the beginning or the explanations of these answers before you move on. At this point, you're going to move through the rest of the practice questions. All right, 5, 6, 7, 8, 9, and 10. You should press play when you are ready to go through the answers for all 5 of these questions. All right, press play when you're ready to go through the answers for 5 questions. All right, at this point, you are pressing play if you've already answered questions 5 through 10. And we'll go over the multiple choice questions. Oh, yeah. Okay. All right, so let's get started on this one right here that's up on the board. According to experiment 1, which cofactor should be used to achieve the highest activity rate? All right, first off, what is the question asking us for? Like which our answer should be a what? A number, a temperature, a symbol, letters. What should it be? A cofactor. What are our three options for cofactors?
Cobalt, zinc, and manganese.
Manganese. Good. So it's one of these three: cobalt, zinc, or manganese. All right. Now, which one of those three will give us the highest activity rate? Cobalt. Good. Yeah. How do you know?
Because I looked at the numbers and it was
Excellent. So these numbers are all higher than any of the other ones. The answer here should be cobalt because it will give you the highest of all of them. All right. Question 6. According to experiment 1, if you did not want folidase function, which cofactor should you use? Now again, what should our answer be?
A cofactor.
A cofactor again. So our same three options we had before. But this one's a little bit different. We want it not to function. How do you get it not to function?
by having nothing no activity.
Excellent. By having no activity. So by having zeros. Now what's the only one that will have no activity across the board?
Zinc ion.
Zinc ion. So this one right here, you notice is zero activity across the board. So Zn2 plus should be our answer here. All right, let's take a look at question 7. 50% enzyme activity would occur at which of the following temperatures based on experiment 2? All right, so in this case, we're looking for an enzyme activity of 50%. And we're looking for an answer that's a temperature. All right, what temperature will do that?
74 degrees Celsius.
How do you get 74?
Because I looked between 70 and 80, and it was 42 to 60%. So in between those numbers was 50%. So it has to be like 74, 75.
Perfect. So 50% enzyme activity, percentages are here. 50 would fall in this range right here, somewhere between 42 and 60. That means that we also need then a temperature that's in between these two, probably a little bit closer to the 70 than the 80, because it's somewhere between 42 and 60. So what did you say, 70, 74? 74 degrees Celsius. All right, question 8. According to experiment 3, one would predict an enzyme activity level of 60% at which two pHs? All right, now our answer should be a pH or an activity percentage.
pH.
A pH. So it's saying one would predict an enzyme at which two pHs. We're actually going to get two of these. All right, we need two pHs. But we're looking for where the activity level would be 60. Why do we need two of these? Why is there going to be two of these and not just one of these?
Because as the pH level increases, the activity increases, but then decreases.
Ah, perfect relationship analysis. As the pH increases, the activity level at first increases, but then it decreases. So that means we're actually going to cross over 60 twice on the way up and on the way down. All right, so where would the first one be?
At 6.6.
Yeah, because it would be really close to here. All right, we know what 59 is, so if we did it over, it would be probably about 6.6 as our first pH. And then where would the other 60% be found?
7.5.
7.5. So it would be somewhere between 94 and 24. Be about 7.5. All right, now this range is huge here. So trying to get an exact number from this side would be very, very difficult. But at least you could estimate that it's somewhere between 7.2 and 8. We're just guessing somewhere around 7.5. All right, question 9. What temperature should you run a pH 7 experiment using 1.2 millimolars of cobalt ion in order to give you the exact same enzyme activity rate as though you completed the experiment at 100 degrees Celsius and a pH of 7.2 using 1.2 millimolars of cobalt. Now, this is where these questions start to get complicated. There's a lot of words here and a lot of variables. All right, but we just need to find the experiment that matches the one that we're looking for. All right, so you should start actually at the end in this case. All right, you should start where it says you're trying to find the same rate as. All right, so you're trying to find the same enzyme activity as though you completed the experiment at 100 degrees Celsius, pH of 7.2, and 1.2 millimolar cobalt. Where would you find that? And I'll say, you don't have to combine multiple ones, you only have to find it once. Where is the number that has 100 degrees Celsius, a pH of 7.2, and 1.2 millimeters of cobalt?
The temperature and table 2.
Now, how do you know table 2? So we can find the temperature easy because it's right here. But what about this other part, this pH of 7.2 and the 1.2 millimeter cobalt?
And the pH.
All right, so now what we want to be careful of is that we have to think back to what experiment this is. If we go back to table 2, that's experiment 2. And in experiment 2, we go all the way back. All right, it says that experiment 1 was repeated, but what temperature was experiment 2 using in this case? Was it constant or were they all different? They're varied. So the temperature varies here. But what was kept the same here? Right, there was stuff that was held the same. Good. The 1.2 millimeters of cobalt, that was the same through the whole experiment. What else was the same in experiment 2? If experiment 1 was repeated, the trials and the 1.2. Yeah, good. The 1.2 millimolar CO2. What else was the same? All right, the pH is actually the same. All right, because it says that experiment 1 was repeated. Experiment 1 had a pH of 7.0. If you repeat that, then that means experiment 2 must also have a pH of 7.0. So in experiment 2, the cobalt ion was 1.2 millimolars. The pH was 7. That's how you can take a look at a question like this and see, okay, we need 1.2 millimeters of cobalt. Already got that. Experiment 2, check. A pH of 7.2, where would we have to find that? Where do we find, because the experiment 2 pH is 7. Where could we find a pH of 7.2? Yeah, in experiment 3, right here. But what about the temperature of experiment 3? Is the temperature of experiment 3 100 degrees Celsius? Yeah. And here is held constant at 100. So in fact, if we're looking for 100 degrees Celsius, pH of 7.2, and 1.2 millimeter cobalt, the number we want is this one right here. Because this whole experiment is 1.2 millimolar cobalt ion, and this whole experiment is run at 100 degrees Celsius, so then we can find 7.2 and find our number. So our target number then is 94%. But the thing is, we're not done yet. We don't want to know the activity level. We want to know our temperature. That you should run a pH 7 experiment with 1.2 millimeter cobalt.
So don't we look at table 2 and look at taking both hot and cold, so I accidentally.
Excellent. Perfect. Exactly. What we do is we go back to table 2, because in table 2, we have the pH of 7.0 that we found out from before. We know it's using 1.2 millimeter cobalt, so that's good. But we can find the temperature that will give us the same activity level. We know we're looking for 94% from over here, so we just trace it over and figure out that the temperature should be 90 degrees. Hardest question on here by far. All right, work. All right, last but certainly not least, according to from the entire experiment, what cofactor concentration, temperature, and pH should you use to achieve the highest activity percentage for proteins? So we're looking to max out every single value to make it as big as possible. Let's start off at the beginning with the cofactor. Which cofactor did we find is the best? Cobalt. Cobalt. All right, these numbers are higher than any other numbers, so we want cobalt. Now, what about that cofactor? Check. We want cobalt. What about cofactor concentration? Which concentration of cobalt will give us the highest? 1.2 will give us 100%. Boom. So we want 1.2 millimolars of cobalt concentration. Check. What do we need next? We need a temperature. What temperature will be the best? 100 degrees. The highest one is right here, which is a temperature of 100 degrees. Last but not least, what about a pH? 7.0, because that gives us 100%. Boom. That's everything. Because you're looking for the cofactor, cobalt ion, the cofactor concentration, 1.2 millimolar. You're looking for the temperature, 100 degrees Celsius, and you're looking for the pH, 7.0. Boom. Okay. Last but not least, this is a question that was actually from a practice ACT. Human blood typically has a pH of 7.4 and a temperature of 37 degrees Celsius. Based on the results of experiment 2 and 3, would prolinase produced by Herococcus gyratusis function as an enzyme in the human blood? All right. So would our enzyme that we're looking at, all right, and this is actually the same enzyme as proteosis. All right. Would this enzyme work in human blood? What do we think? All right, so what do we think? If we're trying to figure out, human blood typically has a pH of 7.4 and a temperature of 37 degrees Celsius. Would those two values work with our bacteria?
No, because 37 degrees Celsius, it would not be active at a temperature lower than 40 degrees Celsius.
Fantastic. We already see as it gets colder, our activity is reducing to where at 40 degrees, it's already 0. So 37 degrees would already be up here, which is beyond 0 already, and we wouldn't have any activity left. The pH part works fine. 7.4 is somewhere in here, and we still have some level of activity. But the temperature is too cold. So then if we take a look at our answer choices, we can already get rid of B and D, because those are both yeses. So we're either left with no, the trend shows that prolinase would not be active at temperatures lower than 40. Or no, prolinase would not be active at any of the pHs listed between 7 and 8. And so in this case, we want to go with which letter choice? A. Fantastic. Because the pH part is fine, it's the temperature part that's bad. All right. If you've successfully worked through this incredibly long video, gotten all of these answers correct, and you feel like you're ready to go, it's quiz time. All right. Good luck when you take the quiz. Check your answers if you get them wrong.